Thank you. Thanks to Abhishek and Professor Shobha Ranganathan for inviting me for this very interesting seminar series or conference. And today I'm planning to talk about how big data can be used in a particular feature of proteins and how it can be applied in a predictive mode. And for this, I take the example of protein domain swapping. So what is 3D domain swapping? And this was observed when first, so that could be the question. And domain swapping is a particular form of protein oligomerization where there is this sort of a handshake movement where a part of the one monomer will intricately start interacting with the other part of the monomer, and this is a special form of dimer formation. And mostly these are homo multimers. And the first recorded example of domain swapping was from Professor Tom Blundell's lab, and it was observed in the beta crystallines, and whereas the gamma crystallines which are shown underneath form normal dimers, the beta crystallines form domain swap dimers. And sorry, the gamma crystalline is shown on top in A. And however, this term domain swapping was coined by Professor David Eisenberg when their group encountered this vivid structural transition, as it were, in diphtheria toxin. So they referred to this as domain swapping. And in this, one can envisage that the normal monomeric state undergoes structural transitions, huge conformational transitions, such that the part of the monomer that is now destined to form now strong interactions with the other protomer will be disengaged, like it happens in satellites and all. So a little part of it is just thrown out in the air, and soon, and when the environment conditions are right, it will form dimer, but now it will form this sort of a domain swap dimer. So this has been nicely described in this protein science paper many years ago by David Eisenberg. And just to recall, these were the people who coined the term. And I'm just going to quickly take you through some definitions because this will help with our discussions on the domain swapping. So this is a schematic representation. The two monomers are shown side by side, not yet engaged in swapping. And what we show on the right hand side is after domain swapping has occurred. So we refer to the little part that has gone from one monomer to the other as the domain, the swapped domain. That is, the little region is the swapped domain, right? And which linker gives facilitates this structural transition among the many linkers that could be in a protein, globular protein. It's a special loop region which we refer to as the hinge region, right? And this hinge is special because it facilitated the transfer of this swap domain to the other monomer. And thirdly, we talk about the newly formed interface that is across the monomers, where the bigger subunit and the smaller subunit of the other protomer are interacting as the swapped domain interface. Swapped domain interface, yeah, please remember this, as opposed to the regular interface, which is called as a non-swap domain interface, NSDI, which will be there in any dimers or homodimers or homotrimers. This is not special, but this is new and very special to the swapping phenomenon. So 4 terms I'll be using again and again. So let us ask what are our questions. Okay. So we asked a question, since domain swapping is not only structurally interesting, but imagine this does not stop with dimer, and it goes on to trimers and tetramers, and in an unlimited fashion, like Professor Shobha Ranganathan mentioned, it's almost like the protein is now lending itself to become unfolded, because then it can, due to swapping, engage in unlimited oligomerization, and it's no longer fun, not even for the structural biologists, because such unlimited oligomers are usually implicated in neurodegenerative diseases. And hence, there is a strong interest to see whether we can recognize even which proteins are likely to form this sort of a swapping event. So we asked this question using computing techniques, which is if we were to implore into the entire human genome can we use machine learning techniques, learning from the known swap structures and applying machine learning tools to recognize which part of the genome and which genes are likely to be engaged in domain swapping. So that is our question. Okay, and for this I'm going to take you through a few steps, and this will form the kind of the logic in the talk today. First of all, if you want to do some prediction, we need data. We need so the positive data set and negative data set. So the first part will be about the data curation and the little analysis, that the structural analysis that will be required to convince ourselves that indeed by knowledge-based method, by looking at the known swap structures, we will be able to apply it in a predictive scale. So the next part is to take you through the machine learning algorithms that we had applied, and then our application on entire genomes. Entire genomes, not only human genome, but a few others, and I'll tell you why. And last but one, once we have large data sets, huge data set that we trust, we can try and do what is called as a meta-analysis. Meta-analysis is like kind of interleaving other related information such as human disease information, human genetic information, because we know that especially when we look in the human genome, our interest is not only the structural aspects, but how certain genes might be involved in neurodegenerative diseases, right? So for that, meta-analysis becomes very natural. And finally, I want to leave you with the thought that are we able to pinpoint which part of the globular protein, namely the gene product from the human genome, and which linker is likely to be the culprit, because that is the one that acts as the hinge. So I would take you through these steps quite one after another. So the first part is to curate the data set. So for this, we started from the entire protein data bank. At that time, there were, this is C, you can see this is 2005, but then we keep updating this database. So what I'm showing the methodology alone, the flow chart. So we start from the entire protein data bank. Of course, we can make it non-redundant so that we are not looking at the same protein at different crystallization states and so on. So that gave rise to about 36,000 structures. And then we apply a domain recognition algorithm that has been developed in-house, which is called as DIAL, Domain Identification Algorithm. And that will help us to know from objective methods, such as measurement of distances between secondary structures and the loops that connect them, and clustering these elements on the basis of these average distances to then recognize spatial clusters that should correspond to structural domains. And in doing that, if we encounter cases where a domain in a PDB contains a bit of the A chain and bit of the B chain, then it would suggest to us that there is swapping going on. So this information has been exploited to pool all those entries in the protein data bank that could form, that are evidence to already have the domain swapping in place. And in addition, just as filters, we make sure that there is a symmetry, because what happens in one protemer should also happen in the other protemer. So therefore, we should expect some amount of symmetry. And we also had done text mining to make sure what the crystallographers think about it. Not that it is final, but if they have also recognized it, we add that value. And that means we do a lot of manual curation. And from 36,000, we were very strict with ourselves, and we created a positive data set, which consists about 2,000 domain swapped entries. So you might be wondering, what is the negative data set? And usually in machine learning, and when it applies to protein structure prediction, always the negative data set is pretty tricky, because when we say these protein entries have been evidence to form domain swapping, we can never ever be sure that those other proteins which are not showing evidence of domain swapping, which will never form swapping. So what we see is only from a crystal structure, right, in a particular pH and a crystallization state. So the same protein, it has in theory the possibility to engage in domain swapping given a totally different condition, a crystallization condition which is different, or the pH may be different. So therefore, when we create a negative data set, we are just making some safe assumptions that such proteins that are not anywhere related to the positive data set members at a superfamily or even at the fold level. So those will form our negative data set. So we curated these data set, and then that kind of shows how you can use near automatic methods to do, so that we can keep updating our database. And this is all present in a database called 3D Swap. And 3D Swap you can see in from our lab webpage. And our lab is fondly called as Computational Approaches for Protein Science, or CAPS in short. So you will find this as 3D Swap. And in fact, as I mentioned, we have been updating, so there will be a 3D Swap Plus as well. So you're welcome to go through this. However, in the present context of structure prediction or domain swapping prediction and being able to apply on the whole genome, this forms the positive data set. And we discussed the negative data set, which are proteins that are in the nowhere belonging to this fold. So then we will swiftly go through what sort of parameters which will go into the machine learning algorithms. And for this, we performed some structural analysis just to be convinced ourselves whether indeed the domain swap protein structures, the positive data set members, are they anywhere different? Are they behaving different structurally from the negative data set? And for this, we primarily concentrated on the hinge region, that is the linker that is responsible for swapping, the swap dimer region, and also the swapped interface, as well as the non-domain swapped interface that I introduced you right in the beginning. The domain swapped interface is the special interface that has been formed after swapping, whereas the NDI is the non-domain swapped interface, the regular interface that is there in general homodimers. And you can see we had looked at features, structural features such as protrusion index, whether there are disulfide bonds, whether the interactions are different, and what about the van der Waals interactions and the hydrogen bonds at the domain swapped interface versus the non-domain swapped interface. And I have put arrows to quickly highlight to you that certain features are dramatically different between the two. The domain swapped interface, it will be interesting to know, although these are newly formed interfaces across the protomers, is not a transient, it's not by chance, because you can see that there are 3 times more van der Waals interactions at this little part that is engaging in interaction. So it seems to be a very purposeful one. And even the hydrogen bond interactions are more, and these are recorded in using our energy calculation method, which we had imparted into a related software, which is called as coil check from our lab, which will primarily start to worry about what are the energetic parameters at a protein-protein interface and dissect them into van der Waals type and so on. Okay, so we are convinced that indeed the positive data set and negative data set are different, but how can we convert it in a way that given an amino acid sequence, we can recognize them? And for this, we have to look at not only backbone conformations, but also amino acid type. And in terms of backbone conformation, interestingly, what we have in the left is a domain swapped region, the hinge region, and you can see that many more observed allowed Pfizer values are there in that swapped region. So again, it is not a chance interaction engaging into some energetically unstable states, because it seems they are, in fact, that there are more allowed regions in the swapped region compared to the non-swapped region. And this again shows you that these hinge regions are special. And how special are these? I'll show you in the next slide, which I hope, because this is stopped moving. I don't know why. Maybe, yeah, now it's okay. So what we have in general hinge, the monomeric hinge, are these reverse turn sort of structures. But what we see in the oligomeric hinge region, where there is a swapping going on, it is like an S-shaped structure. And they have distinct Pfizer values, and I would not be able to go into the details, but just it kind of encourages us to think that indeed the van der Waals interactions are different. The hinge conformation is also different. And what about the amino acid preferences? So here again, this is a very nice table. And unfortunately, since I'm interested more to show you how this applies to human genome, I'm going to go through this a little rapidly. But what we have is an amino acid propensity from our positive data set, which is merely we are asking how often do you find, say, lysine in the hinge region. And this is, if it is says 0.025, it may suggest that it is small, but we are actually comparing it for the general preference of the lysine to be in loop regions. So we are comparing the amino acid propensity of K, lysine, to be in general in loops in comparison to how it is in hinge regions, which is a special loop, right? And we see, therefore, positive values means it has a higher propensity to be present in the hinge, and it has been arranged in the order of preference. So if you see high values, that means it's not a chance preference. It's not as though it likes to be in any loop. It prefers to be in hinge regions. And you can see lysine, tyrosine, valine, glutamine, phenylalanine, and so after that, proline has a slight tendency to be in the hinge region, but most interestingly, residues such as alanine and valine are present more at the hinge region. And what we have shown in these sort of bold arrows in blue and red are the kind of single amino acid substitutions that biochemists all over have already recorded in their papers, and the papers are marked here, and this is from detailed literature survey where single amino acid substitution at the hinge region had dramatic effects. So for instance, there's one example in the arrow shown in red is if a residue, lysine, was present in the hinge region of a domain swapped protein, and if it was altered to glycine, the entire swapping has been disrupted, suggesting that single amino acids can have huge effects. And the opposite is shown in this blue. If it's change a glycine to an alanine, I hope it's alanine because it's covered, but my experience tells me it is alanine, right? So the glycine to alanine mutation, on the other hand, had in fact induced domain swapping. So you see, the kind of system seems to be very suitable for machine learning and prediction of domain swapping. So on the one hand, we see overall van der Waals interactions are more, backbone conformations are special at the hinge regions, and even amino acid preferences are there for certain residues to be present at the hinge region. So with these sort of structural analysis, we went straight on to preparing our system for machine learning. And for this, what we do is to, first of all, finalize our positive and negative data set, as I said before, and create a set for the training, and then we will separately set aside a set for the testing, which will come to. So around 2,000 proteins each were kept from the positive and negative data set for training. And what are all the features? And so we brought in a number of features such as average flexibility index of amino acid, the polarizability parameter of amino acids, the size of residues, and the net charge, their inherent preference to be in the coil region, and so on. So as you will see, almost all these parameters are designs or borrowed such that they don't rely on structural information. You may ask why? Because what we have from genome databases is only the primary structure data, namely the amino acid sequence. So therefore, it makes sense to not rely on structural features, such as we could have easily said, how often do we see residues, amino acids in beta strand conformation? So we could have fed in as parameters, but we have not done that. So bulkiness of amino acids, isoelectric point, all these features have gone into the machine learning. And together with the positive and negative data set, we use the random forest classifier, which allows us to use a combination of decision trees, depending on what sort of parameters can go into the decision. And then together these trees will talk to each other, provide some votes by which, given that there is a positive data set and given that we know that there is a negative data set, the system, the random forest classifier, is going to use and recognize which features are more important than the other, which features discriminate this positivity and negativity, and that is what goes into the training, and then it will be applied on test data set. And if we have any values such as measured by parameters such as accuracy, it will help us to know how well the machine learning training has happened and how well it is working on an independent test data set. Okay, and for this, I have talked to you about the data sets, and I have talked to you about the features, and then I have talked to you now about the classifier, right? So I'm just going to show you a confusion matrix, what as we call it in statistics, which is nothing but knowing that there are about 2000 entries in the positive data set and there are 2000 entries in the negative data set, if the random forest based classifier training is absolutely ideal, you will find 2000 true positives and 2000 true negatives. However, because it is trying to learn from first principles, it is not having the structural information. And what one feature will tell may not always agree with the other. In general, we are doing this prediction with very little information, only the amino acid sequence information. So there are some false positives, some which are wrongly put into the positive data set as though it is domain swap, but actually not true. But the same way, some have left out and they are in fact domain swap structures, but they were treated as though it's part of the negative data set. And therefore, these parameters then help us to know how much is the accuracy, how much is the specificity, and how much is the sensitivity. And you can see that all these values for the training data set of 2000 entries is quite high, around 86%. And the MCC value is a composite information. We call it as Matthews coefficient, which is also quite high. It ranges from 0 to 1. And therefore, it encouraged us to go forward and applying it on a test data set and finally on the human genome, right? And to do this, we next looked at it, how it is in the test data set. And test data set is something that you save it and you make sure that you're not biasing your training. And then the test data set, if your machine learning works very well, that means you are really in a position to apply it on unknown databases, unknown protein sequences. And here again, out of 216, we find that 188 are recognized correctly as true positives. And out of 216 in the negative data set, which we had saved for the testing, 177 of them have been truly recognized. So that again shows you really high values. And for us, it depends on how challenging these questions are. And to give you an idea, I want to tell you, some proteins have signal peptides by which they go into different parts of the cell and localize themselves. And to begin with, the best prediction method available at that time had a accuracy of little more than 50%, maybe 56%, and you generally improve with time. So values such as 80%, or say ranging from 0 to 180.81 values are actually quite good. So armed with the positive and negative data set, encouraged by our structural analysis, and armed with the machine learning classifier and all these features, and knowing that it works reasonably well for a test data set, we then did something which is obvious, which is to apply it on human genome. And we first applied it on the human genome, and we found out of 20,261 genes that we had taken up, nearly 14,000 of them had rendered themselves as domain swapping positive. So nearly 71%, and that looked like a startling number to us because we wanted to know why there is so much response. Could it be true? Or is it just because our method has been overly lenient? It can't have been, because whereas the sensitivity is very high, the specificity, that is how correctly, is it just going to greedily predict many things as positive? No, because our specificity is also around 81%. So we knew that our machine learning is not overly relaxed and picking up many as domain swapping positive. So that means maybe this is how genomes are. And to ascertain and convince ourselves, we applied the same on to other genomes. One is a bacterial genome, and then another is a plant genome. And when we did, we were surprised that similar percentages are seen. Around 70% of the genomes have a tendency to engage in domain swapping. And for us, this was a very good learning and new learning. And we then asked ourselves, if 70% of the human genome are out there to get into a domain swapping, does it make sense from the angle of what we know from disease perspective? After all, this is the human genome, right? And for this, we went on to looking at what is called as a meta-analysis, by which we will like to know how many of them are already known to be engaged in unlimited oligomerization. Is there evidence that they form aggregates? And for this, we found that there are 136 human genes that are recorded to be aggregation positive. And that means maybe they are domain swapping positive. And to our surprise, we found out of 136, nearly 100 of them have been picked up by our machine learning algorithm and training as likely to be domain swapping positive. And out of this, 99 of them are enzymes. So you see that out of the many human gene products that are shown to be aggregation positive, majority of them are catalysts or enzymes, so suggesting that the structural feature of domain swapping might suggest that not only it can form plaques and engage in neurodegenerative processes, but a vast majority of them are tuned, and the signal transduction or the catalytic machinery is kept available for under being of regulation, perhaps through the process of domain swapping. Perhaps the swapping gives them the extra regulatory keys that are required to perform their functions. And then we asked the question that how many of these domain swapping predicted members that we see in fact fall into what types of domains and what sort of enzymes, because we know that nearly 100 of them are enzymes. What sort of enzymes are these? And many of them, in fact 30% of them, are kinases. And it makes sense because we know that several of the kinases are in fact membrane bound, and some are not membrane bound, but then if there are tyrosine kinases and they are sitting on the membrane using a single transmembrane helix, perhaps the formation of the dimer and the phosphorylation, the ligand binding, they're all kind of introduces the feature of domain swapping as one of the regulatory mechanisms. And then you can see immunoglobulins, sugar kinases, and then the pH domains, and so on. And then these are protein-protein interaction motifs, and definitely we see them. So this suggests to us that domain swapping is used by nature not only to form unlimited oligomeric structures, but within catalytic machineries as a form of regulation. Okay, so this is the machine learning part, and then I will like to now stop by saying our challenging question that we had asked ourselves, whether knowing that a protein is likely to form swapping, can we recognize which is the culprit, which is the loop region that facilitates this process. And for this, we had to recognize hinges amongst 3,000 entries, and we definitely can't do this manually. So we set up several structure algorithm in our lab, and some of them are a bit like the David-Rose method of recognizing domains, where we put a plane, cut across a plane on a given protein structure. And this helps us to recognize where are the two domains present, and can this help us to pick up objectively and automatically where are the hinge regions. And for this, since we are testing this approach, we had consulted the literature to look for this term hinge, and where possible, if the residue boundaries are there, we use them. And we found that it is helpful to recognize hinge regions, because you can have loops of all sorts, but certain loops that are crossing one chain to the other and are fairly extended S-shaped structures, as we saw from our analysis, and more solvent exposed. So they are like having more intermolecular interactions, they are more extended, and they are more solvent exposed. Such loop regions were picked up near automatically by us, and we call this as a HID, which means hinge identification algorithm. And we collected a data set, a subset of hinges. And then we used that again, put it through a training, which I'm not going through the details. But then when we applied it on pH domain, which one of the top ranking modules that are there in enzymes, which are known to be aggregation positive, we were able to identify or predict the hinge regions out of this whole globular structure. It has many loops. And out of this so many loops, only this loop was predicted as hinge. And indeed, it is the correct hinge region. It was also shown to be present at the structural transition region, because we do have pH domains which are in the swap form, suggesting that such an approach, being able to pinpoint which part of the structure which has been predicted to be domain swapping positive, could lead to tinkering around, because if we know which is the hinge region, already we know that single amino acid changes can dramatically alter the oligomerization state. And even if we are not able to mutate it, we can identify some small molecules that maybe will sit at that hinge region and disturb the oligomerization state in case the swapping happens to be deleterious for the system and gives rise to neurodegenerative diseases. So with this sort of description, I would like to stop by acknowledging the people who are involved. Prashant did all the structural analysis, like the positive data set accumulation and the HIDE algorithm for hinge, and he's now in Singapore. And Atul Upadhyaya was the brave one who applied it on genomes, including the human genome and later on Tulsi genome. And he's now a faculty in the Punjab University. And we also had previous members like Shamir and Pugalendi, who had actually contributed to the machine learning algorithm. And this helped us to predict genes that may be engaged in domain swapping in the entire human genome. With this, I think I will stop and take any questions which you might have. Thank you.